Right, good afternoon, everyone. Good afternoon, everyone online. I hope you guys can hear me. Guys online, can you hear me? Can you see my screen? The guys online, can you hear me?
Yes, we can hear you, sir.
All right, thank you. Right, so the first thing that I want to do is to give you guys your papers back. The multiple choice that you guys wrote. You guys did really well, by the way. Most of you did really, really well. Right, so I'll call your name. You come and get your paper. As you're coming to get your paper, please take a copy of the paper 3 on that table. Can you hear? Yeah. Exactly my point. They're talking. That's what I'm saying. When you come back, when you come up, you have to get your paper. Pick up the paper 3. That's what I'm saying. Right. So I'll call your name. You come and get your paper. Are we together? Right. Tossie. Toss is not here yet. Maria. Manuel. Shakira. Takiro Chimana. Chance, silent. Chance, not here. Joshua. Tashia, not here. Belinda. Cornelius. Not here. Kapiri. Not here. Shakira Basson. Not here. And Kamwe. All right, so all those people are not here. Is there anyone that didn't get their paper? Okay, let's see. Kamwe. Last I had your paper. Yes. Gina. Uri. Willa. Rasta. Tanwit. Moses. Mamse. Dafa. Not here. All right. Is there anyone that didn't get their paper? Your marks are wrong. All right. Really? Okay. We'll go over the paper. We'll go over the paper. I hope you all brought your question papers. Right. Where should you go? I don't want to be saying the answers. Maybe you can go and write on the digits outside. Yeah. Since you're writing the paper. All right, so please just quickly count your marks, make sure everything is done properly. And then the guys online who wrote the paper one, I'll also give you guys your marks, but then I'll send them individually to you guys. I think Mr. Frank will do that. As soon as we're done this side, I just want to deal with the guys who are here face to face, and then we'll look at your paper. Right, Louise. Okay, so I have to mark that one. Is it the one which was online or the paper on here? Oh, yes. Okay, so I'll just mark it quickly. Right. So, guys, just quickly go through your paper, your marks, and then we'll go through it together. I'll tell you the answers. The answers should be there because I was using a template. So, you should be seeing your answers there. So, quickly just go through your paper. If there's a question that you don't understand, we look at it together. Are we together? Right, so let me just quickly mark here. And then we'll go through it. I hope you all remember to get a paper 3 here. Your paper 3 copy. All right, so let's just quickly share. Where's that paper? The guys online, are you able to see the paper 3? Can you see my screen?
No, sir, I cannot see your screen.
Okay, let me quickly fix that. Can you see my screen now? You should be seeing a paper 3.
Yes, sir, I can see it now.
Okay, thank you. So that's the paper 3 that we want to go over. As soon as we're done with this paper 1, we'll just quickly revise this paper 1 with the guys in the class, and then we'll start with the paper 3. Are we together? Right, have you guys gone through the paper? Are we all okay with the answers? Do you want to go over it? Which questions? Should we go over all the questions or they're just particular questions? Because most of you did really well. Number 16. All right, so let's quickly turn to number 16. So number 16 there, yes, I also made an error there. So you see there's a 6 in front of your glucose molecule, or B. So that means it's wrong. It should be only 1 molecule of glucose with 6. Yes, the answer should be C. That's what I'm saying. If you put B, that is wrong. You shouldn't have 6 molecules of glucose. You should only have 1. I also made that error there. Yes, any other one? Yes. And 30, yes. That was another one there. People were making a mistake. So number 30 says, which sequence shows the correct event in the life cycle of a flowering plant? So with this one, most people are saying it's A, which is dispersal, then pollination, then fertilization. That is wrong. It starts with pollination, right? Then 30, 30. Yeah, so it should start with pollination, fertilization, then dispersal. Are we together? That is the life cycle of a flowering plant. Answer should be C. Yes?
We can see the paper 1.
Oh, the paper 1, it was scanned, but then unfortunately, I can't open it on the PDF. There's a problem with it. I'm not too sure what happened, so I can't share it with you guys. If you actually want to see it, you have to come and pick up a copy because they were scanned and photocopied. Yes, what question? 40. All right, 40 says the diagram shows some effects of human population on the environment. which process is shown in the diagram. So in the diagram, when you've got bacteria that use up dissolved oxygen, plus your water weeds that are growing quickly, fish die from lack of oxygen. That is your C. Eutrophication. Eutrophication. Sometimes caused by your runoff of the fertilizer into the river. Are we together?
Number 4.
Number 4. Okay, let's quickly move to number 4. Right, number 4 says the diagram shows different parts of plants. Which 2 letters show pictures of your dicotyledonous plants? Your answer should be D, L and N. L and N. Right, your K and M, those are monocots. Are we together? The ones that show lateral veins. So the ones that which have the branch veins and the numerous petals, right? Those are your dicots. Are we together? So answer should be L and N, D. Anything else? All right. So if that is done, we can now focus on the paper 3. So we all have a copy. All right, excellent. So if we all have a copy, then we can start the class. Just hold on. Right, so this one, paper 3, this is your alternative practical, right? These are the ones where we expected to know your scientific knowledge, your scientific experiment, how to record data, how to tabulate your data, how to interpret data, that sort of thing, how to get it, then drawing conclusions from that data.
On the multiple choice, what are 6 people?
Okay, back to number 6. Number 6 on the multiple choice is showing your microscope, huh? Your A, that's where you look through. Are we together? Your B, that is to focus your microscope. C is the change in the magnification. You've got your times 10, your times 40, times 100. Then D, that's where the light comes from, right? To light up your specimen, right? Is that okay? Right. Then for this one, let's just quickly go through this paper. Define enzymes. What are enzymes? Yes. Biological catalysts. Protein in nature. That's speed up the reaction. A reaction without being used up. We're together. Without being used up. That is your enzyme. Right. Just 1 mark. But then I feel like that should be a 3-mark question. State what is meant by optimum temperature of an enzyme. What does that mean? When you say optimum temperature here, what does that mean? Tashia and Alpha, you can come and pick up your answer scripts. Yeah, you did really well. Oh, yes, the paper 3, you can pick it up on that table. Right, so let's continue with this question. What is meant by optimum temperature? When they ask you explain what is meant by optimum temperature of an enzyme. What is optimum temperature? The temperature at which the enzyme works. Exactly. The temperature at which the enzyme works best. Are we together? At which an enzyme works best. That's correct. Right, move on to the next part. Now we have part C. Erase this. A lot of interference. Who's that? Who's the participants? Okay, let me just quickly put this here. Right, so part C says catalase is an enzyme which breaks down hydrogen peroxide. Hydrogen peroxide is a common waste product in plant and animal cells. Catalase breaks down hydrogen peroxide to your water and oxygen. Are we together? So if you want to know the chemical formula of hydrogen peroxide, it's what? H2O2. Right, so it's broken down to give you your H2O and oxygen there. Are we together? Are we together? So how would you balance this equation? Let's say if you wanted to balance it. How would you balance it? We've got 2 hydrogens there, 2 hydrogens there, 2 oxygens. So how would we balance it? How would we balance it? How would we balance this one? We've got 2 hydrogens, 2 oxygen. We've got 3 oxygens that side, so we need. We put a 2-way in front of hydrogen here or here, like that. And then what happens? So I now have 4, then put a 2 here. Are you sure? Add the water. Okay. Like that. Then it's balanced, huh? Yeah. If a tissue containing catalase is added to hydrogen peroxide in a test tube, oxygen is produced and foam develops in the test tube as shown in figure 1.1. Right? The height of the foam can be measured and used as an indication of catalase activity. So here, this foam is because of the oxygen present. Are we together? That's forming the bubbles. Are we following? So in an investigation, the effect of temperature on the catalase enzyme was examined using the following procedure. 4 test tubes were set up with 5 cubic centimeters of hydrogen peroxide. Each was brought to a different temperature, 7 degrees Celsius, 20 degrees Celsius, 37 degrees Celsius, and 100 degrees Celsius, respectively, right? Now, a liver cube of 1 cubic centimeter was added to each test tube. The height of foam produced to each test tube was measured after 30 seconds. Are we following? Did we all follow what's happening in that reaction? Now we look at point 1. Table 1.1 shows different variables. Complete the table using a tick to identify the variables that should be kept the same to ensure the investigation was valid. Right, now we want to look at your variables of this experiment. It says the concentration of enzyme. Variable kept the same. Are we keeping the concentration of the enzyme the same? Yes, we are, right? Remember what we are measuring. Look at the experiment, right? We have 1 cubic centimeter of your liver cube, right? And where else? We've got 5 cubic centimeters of hydrogen peroxide for each test tube, right? So what is changing? Anything else that's changing besides the temperature? Is anything else changing? So how about concentration of substrate at start? Yes, keep it the same as well. The pH? Yes, we keep it the same. The temperature? No. Remember, we're investigating the effect of temperature on enzyme activity, on the catalase state. Total volume of substrate in the test tube. Yes. Total volume of substrate in the test tube. Let's go back. Total volume of substrate in the test tube. Let's go to the start. Now, there's no information about that. It should be the same, huh? Since we have 1 cubic centimeter of the liver, 5 cubic centimeters of hydrogen peroxide, yeah, they should be the same. The substrate is, where is it? The liver, is it? The one that contains The hydrogen peroxide. No, liver contains what? What is the liver containing? What is the liver containing? Your enzyme. This contains the enzyme, the catalase. Are we together? Come again? What is the question? The hydrogen peroxide. Remember, we're breaking down hydrogen peroxide. We're breaking down the hydrogen peroxide to water and oxygen. The substrate is the hydrogen peroxide. Are we together? So the catalase is the enzyme that will break down the hydrogen peroxide to give you your product of water and oxygen. Are we together? All right, so let's quickly move on. So this total volume of substrate in the test tube should also be kept the same. Identify the substance in this substrate in this investigation. I've already given you the answer, right? It is hydrogen peroxide. Hydrogen peroxide. The hydrogen peroxide. Right. Then we move on. Part 3. Suggest and explain the effect on the height of the foam if the cubes of liver were cut into smaller pieces. What will happen to the activity if we have smaller cubes? What will happen if we have smaller cubes? Anyone who knows? What's happening when you're using smaller cubes? The surface area, right, of the enzymes. So what happens? More foam is created. Are we together? So you've got, if they're asking for the effect on the height of the foam, what do we say? If we using smaller cubes increases slices, increase the surface area of the enzyme. Surface area of enzyme. Therefore, reaction is faster, right? Reaction is faster, giving more or higher. What can we say about the height? It's higher, right? Hope my grammar is correct today. We say larger or higher, guys? Please check me on my grammar here. What do we say? Higher, right? Giving higher heights of the foam. Is that correct? Sounds efficient. Giving higher heights of the foam. Yes, thank you. Increasing. That sounds better. Increasing. Yes. Increasing the heights of the foam. Are we together? That is the answer. Are you done, Jacob? All right, so I'll mark it during our break. Right, now we move on to part 4. Does everyone understand how we answer part 4 before we move on? Do you want to understand? Is anyone still copying down? The guys online.
Yes, I think the man was calling you now.
His name.
Guys online, there's a lot of chatter there. Right, so we can move on, right? Okay, quickly copy. JJ, where did you put your paper? Okay, I see. All right, so I'll just try to mark it quickly whenever I'm free. Okay. Right, are we done? Yes. Yes, there's still papers. There are still papers. You don't have a copy. There you go. One more. That pile is gone. Is it finished? Okay, so I'll have to ask for extra copies. I think those are the remaining ones. I should keep it low. Really? Means I'm louder. Right. You're looking for the answers. Right, so can we continue, guys? Can we continue? Right, now we move on to part 4. How could the investigation be modified to provide more accurate results? How can we make this better? Repeat the experiment. Okay, repeating the experiment, how? Doing it. I know what repeating means, but then anyways, which part are we repeating? You want you to explain which investigation can be modified to, can we just say repeat? Huh? Can we just say repeat? Can we just say repeat? What can we say, guys? Remember, if we go to the investigation, that's the front of the page. Where is it? Yeah, hold on. Let me just go to paper. There we go. The question paper. Oh yeah, we need extra copies. So I'll ask it to extra copies. Right, guys. So here, as you can see, we are producing what? Water and oxygen. This oxygen, you can measure the volume more accurately. Are we together? Are we together? You can measure it more accurately here. So you can just simply say, measure the volume of oxygen produced over time. Are we together? That's how you can get that mark there. Right. So how do you measure it? You can say with a syringe. Okay. Gas syringe. That's how you can collect the gas there. Right. Are we together? Does that make sense? Right. Let's quickly move on to the next part. Have we all copied it down? Copy it down. How is it work here? Which one? Syringe. S-Y-R-I-N-G. So let's quickly move on. Part 5. Prepare a table in the space below that could be used to report the results of this investigation. Leave any cells blank where data is not yet known. Right, so we want to be able to report results for our experiment. What do we do? What do we do? So remember, what is changing? What is temperature? Come again? Yes. So when you create your table, you can say temperature. Right, don't forget your units. And then the height of foam, right? Height of foam. You can measure in what, centimeters or meters, right? Whatever you want there, right? Since they ask for cells, we have to put the data that we know. What do we know from the experiment? Temperature, right? What is the first temperature? 7, 18. Did you say 18? 20. Okay. What's the next one? 37 and 100. Right. So whatever you get there, for your 7 degrees Celsius and the height of the foam, you put in these cells. So since we don't have this data, we do not include. Are we together? Does that make sense? Where the values coming from? In the table that we were given, or in the passage, yes, yeah. They told us that they were investigating the effect of temperature on the enzyme activity. So we're getting this your 7, 20, 37, and 100. Right. Oh, sorry. You guys are still copying. Okay. All right. Are we done?
So we just put stars on the height.
No, you don't put stars. I was explaining that here you don't put any information because we do not have that information, that data. It was not recorded. It was not given to us. So we can leave it empty. Are we done? Mhm. Right. So if that's done, we move on to the next question. Yeah. That's the table. Is there anything else that we need? No, there's no additional question.
3 marks. So that's 3 marks.
Yeah, it's 3 marks. One for the two.
Put a
There's
line
one
on the
for
height.
this column, second one for the height, and then the data here.
So I'm talking in the sense of 3 marks. 3 marks.
Oh yeah, 3 marks. 3 marks. Yes. I think always try to include the independent variable stuff. The one that you control to the left, the one that started. Meaning whatever you're measuring to the left, I mean to the right. What am I saying? Yeah, to the right. Are we together? Yeah. So can we move on to the next question now? Right, number 2. We've got black currants shown in figure 2.1. A small round edible black berries. Black currants can be ground up into juice containing sugar. Right, so you've been given a picture there of your black currants. I'm sure most of you have eaten those, huh? You haven't eaten them. They're really nice. Or blackberries.
Oh.
Yeah, they're similar to those.
Like the blueberries.
Yes. Right. Now, in an experiment, learners measured the change of mass of potato cylinders in different concentration of black currant juice. They set up 6 test tubes, each, right? So I'll just highlight the important information that we need to take. Each containing 30 cubic centimeters of different concentrations of the black currant juice. Table 2.1 shows volume of the black currant juice and distilled water needed to make 10 cubic centimeters of different concentrations of black currant juice. Right, and that's that table that was given there. Complete table 2.1 to show how to make 30 cubic centimeters of different concentrations of black currant juice. How do we do that? Right. How do we do that? There's already a pattern. So here can you see the percentage of concentration of black currant juice is 0, volume of black currant juice is 0. What is the distilled water? 10. So what is that concentration of that? What's the concentration? The concentration here. The first one. Isn't one making different concentrations? Is it 2?
Yes. So you look at the first.
So here will be 2. Okay. And then here will be 6. Okay. Yeah, I see what you did. Right. Now, how do we now with the distilled water? 0? It's still a sequence. Really? So it's 8 here and 2 there. Okay. Yeah, okay. I see that was the fastest way to do it, huh? Yeah. What if it's wrong? No, it should be correct. Because we have 20%. 20% of the concentration. So it should be 2 and then 8. So such that it's 2 in 10. Are we together? Then 40% will be 4 in 10. That's still correct. 60. Yeah. That was the fastest way of doing it. But then you all see how I'm calculating the concentration. You all understand that. Since here it's 0%, it will be 0 of the black current in the distilled water. And then here 20% will be 2 in the whole total volume. So it's 2 in 10. Are we together? That's how you get the 20%, 2 in 10, because the total volume is 2 plus the 8 to give you the 20%. And then 40 will be 4 out of 10. Does that make sense? Yes. The distilled water, you just subtract from the total. Yes. To get the 8. Yes. Yeah. But then, since you guys noticed the pattern, you guys were able to find the answers. Yeah. So that's very good that you guys were able to do that quick. Now, the learners cut cylinders from the same potato, trimmed the potato cylinders to the same length, right? They used the same length of the potato cylinders. The learners blotted the potato cylinders dry with a paper towel and weighed each one separately. One potato cylinder was placed into each of 6 different concentrations of blackcurrant juice as shown in figure 2.2. So that is our figure 2.2. Yes? Blotted as in you slowly dip the paper onto it to remove the excess water. Like, you know how people apply makeup? Like you're blotting your paper. So that's how you blot. Right. So with this question, what are they asking? Right. So we've got our same potato cylinders of same length in each of different concentration, right? So I think you guys already know what's happening here. Can we assume what's happening? So we know since this one is got 0% blackcurrant juice, right? What's going to happen? What's going to happen? Water will move in. Since there's a higher concentration of your H2O here. So it will hypertonic solution. Thank you. This is a hypertonic. Hypertonic. Right. Moving to hypertonic solution. No, there's no 50%. Right. Now, after 30 minutes, the learners removed each cylinder and blotted it dry. Each cylinder was reweighed and the mass recorded. Table 2.2 shows their results. All right, so let's quickly look at the results now. So your 0%, you can see the mass of the cylinder at grams, it's 8.2 grams. At start, yeah, 8.2 grams. Right? And then mass of cylinder after 30 minutes is now 9.15. Change in mass is 0.95. The percentage change in mass is your 11.59. Are we together? Can we all see that data there? Now, they ask you calculate the change in mass and the percentage change in mass of the potato cylinders in the 100% concentration of the blackcurrant juice. Record these values in table 2.2. Who can do that for us? 4.95. Negative 4.95. What else? How do we get the percentage change? Come again? Negative 55. What's happening? That's the answer. I'm asking, how did you work it out? How did you work it out? Yes. Come again? What's wrong? For here. Over 8.95. It's negative because it's losing. It's reducing in size. The change in mass, it's losing. Are we together, guys?
5.39.
All right, let's go back to this one here at the start. At 0. Wait, hold on. Confirm here. You just simply took the 9.15 and subtracted 8.2. Are we together? That's how we got the 0.95. Meaning you should do the same for everything. So it's your 4 minus the 8.95. 4 minus 8.95. What do you get? Yeah.
Right, sir.
So it's correct.
55 is 55.3.
3-1. Okay, so how do we do this one? All right, let's go back to the first one here. Here, the change in mass, the percentage in change in mass, you take this, right, over the what? At the start, right? Then what did you get? Is it 11.59? Is it 11.59? The 0.95 divided by 8.2. It is, right? So please do the same thing here. Minus 4.95 divided by 8.95. Minus
3-1, not 7-1.
3-1. You're getting 3-1 now. Okay, so we're all on the same page now. Okay, 3-1. Does that make sense now? All right, guys online, are you with us? Are you following? Come again? Oh, you can't. I'm not too sure. I'm not too sure. I think he might be passing by church first and then he comes to the side. All right. The guy, what was that? Who's the Shigweda? Is that how you say the name? It's a new student. I'm not too sure. Shigweda. Shiweda. Guys, I don't know how to pronounce names. Okay. Shiweda. Right. With the calculation, for us to calculate the percentage, you understood how we calculated the change in mass first, right? Just indicate in the chat box if you understood how we calculated that part. Okay. So now, for us to find the percentage change in mass, you first take this change in mass, right? The minus 4.95, and divide by the original mass at the start, which is this one here. So it'll be divided by 8.95. Right. And then since you want to change it to a percentage, you multiply by 100. That should give you your answer, which they're saying is minus 55.31. Are we together? Does that make sense? Together? Okay. All right. So we're all on the same page now. Right. So can we move on now? Okay. Right. Oh yes, and again, guys, before you move on, please make sure your results are consistent here. If there are two decimal places, your answer should be 2 decimal places, as well as this column there. If you just leave it as 1 decimal place, you get it wrong.
So they don't specify.
They don't specify.
Yes.
No, they won't specify. You just expect it to know that you have to keep the results consistent. Yeah, because no, it makes the experiment fit. You can't use 1 decimal place, 1 place, and then use 2 decimal places with the other. It skews your results. Right, we move on to the next part. Part C. The learner's cut cylinders from the same potato. Suggest one reason why it is important to use the same potato. Come again? Yes, to make it fair, right?
Yes, to make the experiment.
Yes, to make the experiment fair. To make experiment fair. Are we together? Does that make sense? Right, so remember, different potatoes can have different sugar content in it. Are we together? So you have to make it fair. The different potatoes Potatoes have different sugar content. Different sugar content. Yeah, I'm just giving one point that you can also include there as to why you use the same potato. Yeah, you can use it. This one is still correct, right? But then this one is explaining further. It's more detailed altogether. Right, Cornelius, you can come and get your answer scripts. Hold on. 4. Right, let's move on. Are we all together there? Right, so let's quickly move on to D. Use the data in table 2.2 to plot an appropriate graph of percentage change in mass against concentration of blackcurrant juice on the grid provided. Right, so with this one, I think we looked at some of the questions with the category of K. I want you to tell me which one is going on my y-axis, which one is going on my x-axis. I wish I had something that I can draw a straight line, but I don't. Where's the ruler? No, it's on the iPad. It's an application of the app. The pin's on the. Oh, this. Does it allow it? Okay. It doesn't. Actually, HTML. Right. So can we just quickly draw that? As I'm doing it here, you guys can also be doing it in your. So we said here on the x-axis is the. What is it? Concentration of the blackcurrant juice. Concentration of blackcurrant juice. And then on the x-axis is percentage change in mass. Percentage change in mass. Are we together? Right. What scale are you guys using? What scale are you using? If you look at the result page. 0.0. Which one, sir? Okay, let's start with the x-axis. X-axis, we know our percentages were going from 0 to 100, right? The concentration was 0 from 100, right? So you can say from 0, you can say 20, 40, 60, 80, 100, right? So 5, 20, 40, 80, 100. That scale is 5, right? Cover more than half of the grid. Now with your y-axis, let me just quickly zoom out. What scale are we using? 0.05. Let me look at my table. Percentage change in mass. Here we've got negative values. So our 0. -5. Let me remove this 0. So that doesn't confuse us. -5. -5. -5.
Nah, it should be -10.
-5.
Maybe negative.
Right. So can you see our biggest value here? The biggest is 11.59. Then the smallest is -55. Yes, that's 55. 55. So what scale can we use to cover that range? So remember, guys, since our negative values here, that means this part will have to be where? It will have to be somewhere here. Let me quickly erase these values. Since we are dealing with negative values, we'll have our x-axis coming in between. So if we have 11 here, we can put it here, since most of the values are under 0. We can't put them there, we have to put them here. This is our 0 mark, since we've got negative values all together. Does that make sense? It's complicated. No, you'll see. Because we've got positive values and negative values, we need to show the difference. So this will be your, what do we say is the scale? Can I use 10, 20, 30, 40? 10, 20? Can I use that one? 10, 20, and then this will be minus 10, minus 20, minus 30, minus 40. It doesn't work. It doesn't work, right? It does not. So let's try again. One above. As in put my 0 up here. Yeah, that can work. Yes. Yes, that works. That works. You'll see. Right. So here, if we raise it above to the middle mark here, you'll see. So now, this is the x-axis, right? This is the 0 mark. This will be what now? What will this be? This will be 5. Are we together? Then the next part will be 10. Then the next part would be 15. Does that make sense? It is the same. What do you mean? Here and there.
So we pull down the 5.
Yes, they'll be the same. It's just that now I've moved the line to the 5 mark. Instead of using the 10s, 20s, 30s, I'm using the 5s now. Are we together? So now I'm using 15 there, 0. Then this will be minus 5. So I'll just quickly write my minus 10 there.
Sir,
Yes.
can't such as like do it like positive 10 up.
10.
Instead of like a get making it 5.
Uh-huh. So I just say positive 10 here.
Then the 5.
At the 5, I put 10 here.
Yeah, because the negatives will like, I don't think it will reach negative 50.
Okay, fine. Let me show you something. If you use 10 here, this will be 20, right? Then this will be what?
Negative 10.
Negative 10, negative 20, negative 30, negative 40, negative 50. So you can see it's not covering a lot. But then I think it'll still work. Negative 20, negative 10.
But the negative 5 might not work.
Okay. All right, so we want to use this one, the negative 10. Which one suits better? Let me see this one here. The plus 5, the plus 10, plus 15. Where's my middle line? Minus 20, minus 30, then minus 40. So it doesn't work, huh? Okay. So let's use yours. Yours works. Right. So we put our 10 here. What did I do that? Right, so this is 0. So I'm saying I put my 10 there, right? What's throwing you off? The 10 points, what? I don't think we can include those because our scale is quite huge. So we can't really include that. So if you have here, this is your 10, 20, and then what else? Minus 10, minus 20, minus 30, minus 40, minus 50. Minus 50 there. So this will be minus 55. then minus 65. Right. Is that okay?
No.
What's wrong?
Why is it changing from the 10 to the 5?
Oh, sorry. I made that mistake there. So this should be 50. Okay, yeah, thank you for that. This is be 60, 70, 80. Right. Right, now we need to plot the result. Huh? Right, so this one, 1159, you can easily put it at 12 for the first concentration. So this is my 20. This is my at the bottom. Okay, but then here I'm just trying to show you that this is the 0 mark. Yeah. So I think you can put that at the bottom as long as you show where the 0 is. Yeah. This is 40, this is 60, 80, 100. So at 0, it's 12, right? At 0. Is it 12? Oh, yes, the percentage. So we put my x there. I'll just try to zoom in a bit to be more accurate. Right? And then 20, what do I have? It's 0. So I put my 0 there. And then 40, 10. Minus 10. Right? 60, negative 6. The negative 6 is up here. It will be 2, 4, 6. Will be there. Right? Then 80, negative 44. So it'll be down here. So at 40, 2, 4, there. Right? Next thing, 55. So 55 will be 2, 4. So in between these two lines, that's where I draw my 55. There. Are we together? What's wrong? What's wrong?
It's going to be straight to the number.
Right, that's our graph, right? What are you saying is wrong? It's just weird. Okay. It's still a straight line, yeah. Yeah. So now, what can we do from here? You can draw the line of best fit, or you can join your points. But I think it's a line of best fit, yeah? Line of best fit. Let's see it. It doesn't state. Usually you can tell, but through the question. Since these are results, we're trying to investigate two investigations. We've got your change in percentage mass against your what? Concentration. So you want to see how does your concentration of the black current change with what? The loss in mass. So you're investigating two things, your correlation. So usually that one, you use your line of best fit, or they connect the dots. But then this one looks like it's a line of best fit, since most of them are together. Can you see that? But then where it's you're looking at the rate of change or something, you draw a curve out together. When you're looking at rates, calculating rates of production of your gas or your oxygen of an equation, you use your curve. So with this one, try to draw the line of best fit. Yes, line of best fit. Where's the ruler? Right. So I'll try to draw the line of best fit. Right. bad drawing. Sorry? Is it okay? It's not okay. It is not okay. Something like that. Something like that. I feel like that one is close enough. Are we together? Yes? Did you all get something like this? Come again? You've got one here and one there. This one, can you see it's different it's compared to these ones, the ones that are close together. We can say this was an error because it's totally unrelated to these ones here. You just draw it. Yes, you ignore it because it's not correlating with these ones here. Can you see that? Even with your table, can you see that there's a different change? It's not correlating, it's not decreasing. So we know this one isn't. This is an error. Are we together? Yeah, that's what I'm showing on that graph here. That's why I'm ignoring this point here. Does that make sense? Did you all get something like that? Who tried to draw the graph? Did you all try to draw the graph? Let's see chance. How did we plot the points? Okay. Hold on. Right, guys, for the ones that don't understand how we plotted the points, here we're using a scale of your every 5 boxes is 10, right? So since we have positive and negative values, our 0 mark is starting at the top there, right? Now I draw since my biggest value is 11 from the table, my biggest value is 11, right? So meaning I can start at the very top here. That's why I'm starting at 20. So 20, the next 5 is 10. So meaning one box will be what? You just divide by 5 to get one box, which is 2. So one box is 2. So you're here, we'll be counting downwards. Your 20 will be 18, followed by 16, then 14, 12, then 10. Does that make sense? So you now understand the scale. Even downwards here, negative 10, negative 20, since the lowest value that we have is a minus 55. So minus 55 will be somewhere down here. We mark this point there, right? Now we look at the different concentrations that we had. At 0, it was 11, right? So we put the mark there. And then at 20, what was the mark? 20 was 0. That's practically 0. So that's why we put it there. So yeah. Oh yes, because our scale doesn't account for all of that. It's too big for us to use the decimal points. That's why we're rounding off. Are we together?
In the value for 40.
40. Value for 40 is, isn't it 10? What did you say the value for? Yeah, 10. 9.56. If you round that off, it gives you 10. So that's why I put it 10 there.
So we rounded everything off.
Yes.
That's why 0.22 is 0.
Because we can't draw the decimal points inside. The scale is just too large to account for everything. Right. So did you all draw the curve? It should look something like that. I mean the line of this fit. It should look something like that. Did we all get something like that?
But ours are
Yeah, not. Yeah. You try and come and draw the board and see how it comes out. Right. So remember, so I'll tell you the mark allocations here. They'll give you a mark for your x-axis, a mark for your y-axis, then a mark for your scales that you used. Are we together? That's how many marks? 3 marks. Then a mark for your points correctly plotted on the graph, and then the mark for the line. Are we together? That's 5 marks. 5 marks for all of that. So please make sure you use your proper scales, and then your axes are labeled correctly, and then you plot your points correctly and draw the line properly. Are we together? You get the mark. Right, now we look at E. E is asking you, suggest the reason for the changes in mass of the potato cylinders between concentration 0% and 80% of black currant juice. What happened there? The concentration decreased. Yes. Decreased. Yes, correct. As the concentration of black currant juice increases, black currant juice increases, the mass of the potato cylinders decreases. Because we use potato cylinders. Decreases. Are we together? So you get a mark there. As the concentration of the black currant juice increases, the mass of the potato cylinders decreases. Even if you look at the graph there, that's what's happening. Your concentration is going up, but the mass is going down. Can you see that? So you can still get that relationship from the graph there. Right? That's your 1 mark.
1 mark, almost the whole thing.
Yes. Right. Any other thing? What can we state? What else is there? Yes. Yes, you can state that. We were saying that in the beginning, there's more water molecules outside, right? So you need to state that the water potential is higher inside the potato than outside which causes decrease in mass. Are we together? So you can also state water molecules move out through osmosis. Through osmosis, yeah. So the reason why your mass of the potato cylinders is decreasing is because of the loss of water through osmosis. Are we together? Anything else? So you can quickly copy that down. Right, so it's important to note that from your 0 to 20%, where there was an increase, what happens? From 0 to 20? Yes, the percentages. So can you see from 0 to 20 there, it's about. Did it decrease or increase? Decrease. It's increased. That's why the positive values are big. Percentage change in mass. It's positive. Go from 0. So they increase in. Yeah, they increase in mass because the water was moving in. But then here, where we have the line below the graph here, where it's negative, they reduced in mass, right? Meaning they were in more concentration. The water molecules outside the potato, what was wrong? Was lower than in the potato. That's why you have water molecules moving out of the potato, reducing the size of the potato or the mass of the potato. Yes.
Because we did not move from 10 to 0.
From 10 to 0.
Yeah, in the first 0 to 20.
If you look at the graph, your table.
Yeah.
Which parts?
From 0 to 20, sir.
Yes.
From 12
Yes.
to 0.
Yeah. There's still an increase there.
Not a decrease.
It is not a decrease because it's a positive value. Decrease means that it's negative. It lost mass. Yeah. So all of the values that are between 0 and 20 were positive. Does that make sense? So that's why we're stating that any value that is above your 20, this part above this line, all of that were increased in mass. The water was moving into the potato. Then everything that's below this line here lost water. Are we together? The potato was losing water, which is a decrease in mass. Does that make sense? Okay. Does that make sense so far, guys? Right. So I hope you understood that experiment. Yeah, but then that's how they usually are. That's how they ask those questions. So always just try to see the relationship. If they ask you to draw a graph, try and see the relationship from the graph. Did we get the answer? All right, so can I move on? All right, so I think we're done with question 2. We can move on to question 3. What is mitosis? Describe mitosis. What happens with mitosis? What is happening with mitosis? Yes, Kopire? Yes, the cell is dividing. It's a type of cell division. What happens? Yes. That's all. What's your guess? Yeah, thank you. This is a type of cell division.
So must you specify the opposite?
Yes, type of cell division that creates how many? How many do we create? 2 identical daughter cells. Yes, that's the identical part. Daughter cells. Are we together? Now they ask you outline the purpose of meiosis and mitosis in the human body.
The fusion of the.
Come again? Diffusion.
No, fusion.
Fusion of what?
Cycle.
No. Yes. We said growth. Which one? Yes. Growth. What else? Repair. Repair of damaged cells. What else?
Duplication.
Right? And then here, what can we put? Yes, production of gametes. You said that's not a zygote. Zygote is when you're actually doing mitosis first. That's why you have numerous cells. Production of gametes. So remember, what is the number, the chromosome number for gametes? What is the chromosome number?
24.
26.
26 or 23.
23. There is 23. 46 chromosomes is your mitosis. They start from mitosis. Are we together? We have 46 chromosomes or 23 pairs of chromosomes. Are we together? Does that make sense? So your haploid number, that's why you have it as N for meiosis and then 2N for mitosis, because they're multiplied twice. Are we together? When you create your gametes, you divide them into 2. Are we together? So that there's no double in every generation. Now let's look at the root tip. Zoom in. Okay. Is that okay? Which part? Okay, yes. Yeah. No, for your level. No. Not for your level. AS level, yes. We expect you to know all the stages of mitosis. So if you're planning on doing biology, then you can look at that. Right, can we move on to the next part? Are we all okay now? Right, now B, the root tip squash procedure can be used to observe cells undergoing mitosis, right? Complete figure 3.1, which describes the stages involved in preparing a root tip squash to show mitosis. Write the most appropriate word or words on the dotted lines. Right, carefully remove the root tip from the iron that has been grown in water. Right, place the root tip in something acid. What happens there? Which parts now? Okay, quickly read there. Yes. Yes. What's the answer? Maria? You mentioned the answer. Yes, hydrochloric acid. Hydrochloric acid. Hydrochloric acid. It's not corrosive. It's talking about we want to remove the cells, to open the cells. We want to open the cells. So the changes in the concentration of the iron to open the cell membrane. Yes? Yeah, to open the cell. Oh, okay, I see. So most of you don't know this. Hydrochloric acid. Come again. So which school did you go to, by the way? Yeah, no. So most of you guys, did you get the paper? You guys didn't get the paper. So you don't know the procedure. Okay, so I'll try to look for the procedure for you guys online. I will. You remind me anyways. Right. Okay, let's finish. Let's finish. I'm pretty sure when we're done with this, you'll know the procedure. Are we together? Since you guys are writing down the procedure. Right? So, which will dish the tissue? What happens to the tissue? I just explained this. Break up the tissue. Or open up. Yes. No, you can't say just open. Break up the tissue. Are we together? Then rinse in distilled water. Then add a few drops of a stain such as acetic or same. Yes, ethanol is same. Ethanol or same. Or same, like that. Ethanol or same. Right. The others you can use are your toluidine, the Schiff's reagent, right? That's yeah. So any of those, if you put them, you get a mark. So choose which one best you can to remember that. They're both types of, they're all types of stains.
So what are those words?
This is ethanol or same. This is toluidine or toluidine, however you want to pronounce that. This is Schiff's reagent. Those ones. So whichever you prefer, the one you can pronounce, you can take.
Schiff's what?
Schiff's reagent.
Another one.
Toluidine. 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 The type of stain.
T-I what?
Luidine. It's for testing for mitosis, the stages of mitosis. It shows them, each of them. On the stages. So here, I want to show you the stages. I want to show you the stages. Right. So guys, so remember here, we're using root tip. It was a root squash, right? A root tip squash. The reason why we're doing this, we're staining the chromosomes. Are we together? so that you see which stage at which each of them are. Does that make sense? So that's why we have to open the cell first, so that you get the nucleus. Yes.
I don't
Eukaryotic cell. Right. Now, stage 4, to prepare to view in light microscope, place the stained root tip on a glass slide. Yes. Then place a cover slip. So you guys have done this before.
I never done this.
Seems like most people know it. How come you don't? Really? Okay, but then now you understand the procedure. Do you understand the procedure now? Should we go over it again? All right, let me try and Google some pictures so I can show Kakuyire. Right. Right, so root tip squash, practical. Right, so I'll quickly show you this picture. Right, so can you see here what's happened? Can you see that? These are the cells that have been stained. I can't write. Those cells have been stained with your shift reagents, your ethanol, or say your solid. That's why we're able to see them under the microscope. The reason why we use the HCl is to break up the cells to open so that the stain can enter the nucleus. Does that make sense? So when it stains, that's how it appears on the microscope, under the light microscope. So can you see that we have some that are, yes, these ones are the chromosomes. Can you see some are already in mitosis? Others are still preparing to go into mitosis. So before mitosis, your cell replicates. It creates more chromosomes, it creates more cells, more organelles, right? And then the microsomes, not the microsomes, is it the chromosomes? They migrate to the center. Can you see the ones that are at the center? These ones here. Then this one here, they're splitting up. They're splitting up. Can you see this one here? The one at the bottom right there. They're splitting up to form the two cells. So it's within that mitosis procedure to create your two identical daughter cells. Yes. Come again? The nucleus. Yes, the chromosomes are the nucleus. The chromosomes are the nucleus. Are we together? That's where you find your genetic material, which is the chromosome. Does that make sense? Are we together, everyone? Can I close this? Online guys, are you with us?
Yes, sir.
All right, do you understand that question now?
The root tip first. Yes, sir.
Yes, the root tip part. The one where
Yes.
the experiment is to show the stages in mitosis.
Yes, sir.
Okay, excellent. So we move on. Stage 4. Quickly copy down. You guys are not listening at the back gate. They are not listening. I'm already talking. But most of us, we are done. So we want to move on to the next question. Please do not waste other people's time. Right. Are we done? So can we move on? Now, part 2. Suggest why the root tip is a suitable part of the onion plant for the observation of mitosis. Why is it suitable? Yes, there's constant growth. Constant growth. Constant growth of your cells. So that's why it is suitable. Are we together? There are cells that are continuously in mitosis. Yes. Yes, it's not that it's about the growth hormones, but then we're looking at the mitosis. You can't really mention the hormone, but then if you mention the growth part, I think you can take your answer. Yeah. But then it's correct, your auxins are found at the roots and the shoots, right? So that's why they're constantly replicating, forming more cells. Yeah. Right, now figure 3.2 shows an onion root tip cell before mitosis starts. So this one I'll have to zoom out. Right, so it's showing you at times 400. Are we together? That's the magnification of that cell, right? Use label lines to identify the cell wall, cytoplasm, and nucleus on figure 3.2. Right, which one is my cell wall, which one is my cytoplasm, which one is my nucleus? Yes, Tashia? No, I'm asking you, you know. I'm asking you if you know which one is the cell wall. You can't see. You didn't bring a glass today. All right, so would you like to answer, Belinda? You don't know what I'm asking? Who's in this class today? Yes? I don't know what the yeah. This is the cell wall. Do we agree? Right? Which one is my cytoplasm? The clear parts. Cyto. Yes. Well done. And then where is my nucleus? Nucleus. Are we together? Yes, Shakira. Come again? You wanted to answer the next question. Okay. So what is the next question? Use the ruler to measure the length of the cell labeled A in fig 3.2. What is the length A? So with this one, I think I'll have to have my pointer make it. So does it show the zoom in? Because it will change the length. Yes. You can use millimeters. No. You can't use millimeters. Right, guys. So how do we find? They're saying use a ruler to measure the length of the cell labeled A in fig 3.2. What measurements are you guys getting? 28. 29. 28. 28. 2.9. Okay. Centimeters. They say 2.9 centimeters or 29 millimeters. No, if you specify, you put your measurements, yes. So 28 millimeters. But I think usually these answers are very exact. Are we together? They are very exact. Are you guys measuring this side? Yes, I understand that. So you're using how? How did you measure the length? From this side, right? Yes, sir. To there. Please make sure you did that. If you measure the other way like this, if you measure like that, that is wrong. Are we together? We're looking for A. So this part to that part, use that. So it's even better that they gave you lines. You can just put your ruler there. Are we together? So your answer is 29. 28, 25. 29. Okay, thank you. So 2.9 centimeters or 29 millimeters. Right, so let's move on. Calculate the actual length of the cell labeled A. Show your working and give your answer in millimeters and micrometers. Are we together? Yes. Yes. Yes, the 2.9. Divide by 400. Wait, 2.9. Divide by 400. Or 2.9. I think you can use anything. I think you can use anything. Millimeters, or they can change it to micrometers. Yes? What is the answer? Just give it to me.
Sir, it says 7.25 times 10 to the power.
22? Come again?
So it's like 7.25.
7.25
That's 10 to the power of
negative 3. What did you use? 2.9, huh? Right, so guys, this 2.9, we use centimeters, right? So for you to put it into millimeters, you divide by what? 100. Are we together? Does that make sense?
Don't you multiply?
So you multiply by
You multiply by 10.
Yeah, you multiply by 10.
Which part?
10 centimeters.
Centimeters to millimeters.
To millimeters. We have minus 3, right? So when you have a centimeter, how much is a centimeter?
10
Oh, yes, 10 millimeters. So we multiply by?
10.
Do we multiply or divide?
Multiply.
Why are we multiplying?
Oh, yeah, you divide. You divide.
Because we
Give it centimeters to
Yes.
millimeters.
Yeah,
Yes.
that's what I'm saying.
So we are converting this into millimeters. So what do?
Multiply.
We multiply
By 10.
with the negative 4, yes. So that's correct. 7.25 times 10 to the minus 4. So your answer here, you put it as
Yes, sir.
what's
Yeah,
wrong?
right. It's right. No, no, say it's wrong one.
Which part is wrong?
C divided, so you multiply by 10.
No, guys. Okay, let's start again. Let's start again.
Yeah, let's just make our lives easy.
You will still have to convert. You will still have to convert this here. This part.
It's fine, sir, but let's start with millimeters.
All right, let's start with millimeters then. What is the millimeters? You're saying 29. So remember, your magnification is always equals to image divided by object. So what is my image? 29. Right? And my magnification is 400. So we're looking for your actual, the object. So object will be equals to your 29 divided by 400. What do we get?
0.07.
0.725. Like that. 0.0725 millimeters.
It's our first answer.
0.0725.
Yes.
Okay, which is C. Okay, so we divide it by 400, which is correct there. You get your mark there. 0 point. Okay, I see. So this will be the millimeters, right?
Yes, sir.
Okay, so 0.0725. Then how do you convert this to micrometers? You multiply by 1,000. Yes. Yes. Right. What does that give you? 72.5 micrometers. Are we together? 1,000 is
Micrometers.
yeah. So remember this, yeah, micrometers is 10 to the power minus 6, and millimeters is 10 to the power minus 3. So you multiply by 1,000, the 3 zeros. So
meter, centimeter, millimeter.
right, let me write it like this. So here, since it's millimeters, in meters, this would be times 10 to the power minus 3 meters. Does that make sense? Does that make sense? Right. For us to get to 10 to the power minus 6, we multiply by 1,000. For those the extra 3 zeros. Does that make sense? So you multiply the extra 3 zeros, you get your 72.5 times 10 to the power minus 6. Yes. Yes. Yes, sir. How do you know it's micrometers? Micrometers here is minus 6. Always. Always. Micrometers is always 10 to the power minus 6. Milli, that's why we know it's 10 to the power minus 3 of meter. Are we together? Same thing for nano, it's 10 to the power minus 9. Are we together? So it's always like that. Those are the denotions for your measurements for mass. I mean, length. Yes, it's always important to know that. Micro is 10 to the power minus 6 meters. Right? So this is your measurement like that. And then this is your millimeters, which is 10 to the minus 3. Then the nanometers, which is 10 to the power minus 9. Does that make sense? Minus 6. Minus 6. Nanometers is 10 to the power minus 9. Are we together? Right, so let's move on. Are you still copying? It's break now. What's the time now? It's 2 o'clock. Okay. So if it's 2 o'clock now, let's see. All right, it's fine. So we can take the break. Since we're finished with number 3, we'll come back and do number 4, and we're done. I think. Then we look at the other paper. Is that fine? All right, so the guys online, we can take a break for now. I think we'll resume at 2:30. That's fine.
Yes, sir.
All right, so see you later. Wow.